Antibiotics were discovered over a century ago, and since then, we've been using them to combat bacterial infection. They play essential roles in practically all sectors of human civilization, and are still one of our most successful factors of medical intervention even today. However, they are also a double-edged sword. Antibiotics pose a selective pressure on bacteria, and such they may evolve resistance. Inappropriate use of antibiotics may lead to a rise of antimicrobial resistance, causing 5 million of deaths worldwide annually, according to the WHO. If we are to do nothing, the related deaths are projected to reach 10 million annually by 2050, making cancer the sixth leading cause of death. Not to mention the global GDP loss of 100 trillion USD alongside it.
Of course, there are methods currently on the market to test for antibiotic resistance. However, they prove to be regrettably inadequate in certain cases. For example, genetic tests like PCR are not very accessible as they require expensive equipment and trained personnel, which is infeasible in low-resource settings, notably in low and middle-income countries. While minimum inhibitory concentration tests are time-consuming since they require lengthy culturing, and routine culturing is only available in 1.3% of labs in sub-Saharan Africa. Unfortunately, 86% of the annual MLR-related deaths do in fact occur in limits. There is an urgent need to enhance infection prevention and control, making affordable diagnosis more accessible in this region. The Lancet series on AMR suggests that enhancing infection prevention and control standards in limits to match higher income countries can prevent up to 337,000 deaths per year. And one of the most effective strategies is to make a quick and accurate diagnosis to prevent antibiotic misuse. Developing such a cheaper, faster, and more accessible system is precisely what we, the ResiSense team, set out to do.
To that end, we present ResiSense, a cell-free point-of-care strategy for rapid detection of bacterial antibiotic resistance gene. With ResiSense, we aim to provide a cost-effective alternative for antibiotic resistance screening by emphasizing user-friendly operation. The mechanism is designed to eliminate the need for dedicated on-site hardware. In the first step of the mechanism, the collected bacteria sample is lysed to release its DNA. Step 2, if the bacteria possesses an antibiotic resistance gene, that gene will be digested by a nickase and a restriction enzyme, which is then followed by linear strand displacement amplification, or LSDA. After nicking the 3' site and cleaving the 5' site, 529 DNA polymerase will bind to the nick site and initiate a DNA polymerization up until the cleaved site, while simultaneously displacing the upper strand. Step 3. The displaced single-stranded DNA now binds to a circular template for kickstarting a 529-mediated rolling circle amplification process. Without the sequence-specific primer, the RCA should not run, which contributes to the detection specificity. This second and third steps are designed to occur in a single reaction mixture. We specifically designed our RCA template so that the amplification product generates a long continuous strand of G-quadruplex structures that can be further split up by the same nickase. In the presence of hemine and hydrogen peroxide, the G-quad functions as a peroxidase mimicking the enzyme that catalyzes the oxidation of tetramethylbenzidine, TMB, indicated by the color change of TMB from colorless to blue. So, in the final step, if the final reaction mixture has a blue color readout, it means that the G-quadruplex structure was produced successfully, which means that the antibiotic resistance gene was present in the sample. For proof of concept, we designed a synthetic part KPC2 surrogate that mimics the gene of Klebsiella pneumonia carbapenemase 2, a beta-lactamase that is responsible for inactivating the last-line antibiotic carbapenem. The part is cloned onto the PSV1C3 backbone, which is then transformed into the DH5 alpha strain of Escherichia coli as the mock-resistant bacteria. Then, we tried to lyse the E. coli cells with SDS at room temperature. Our experiment shows that the number of intact cells is significantly reduced after the treatment, as shown by the red circles in this picture, which means that this particular method of lysing the cell is a viable option for our first step in the mechanism. We also tested our mechanism of extracting the targeted resistance gene from PSV1C3 KPC2 surrogate using anti-BSM-A1 as a nickase and HHA1 as a restriction enzyme. From this LSDR reaction, we expect the displacement and amplification of the 32 nucleotide single-stranded DNA, as well as 724 and 524 nucleotide type products due to the nature of PSP123. This gel electrophoresis result indicates that our LSDR reaction has successfully generated both side products. However, we're unable to detect the 32 nucleotide primer, potentially due to its smaller size, making it appear fainter in the gel results. To test the RCA reaction separately, we utilize a 32 nucleotide synthetic primer for the preliminary testing instead of using the extracted one. In one of our experiments, we tested RCA reactions with different RCA template concentration. Lane 5 over here was set up in a reaction mixture containing both 1 nanogram template and a primer without anti-BSM-A1. The result shows an expected smearing band with sizes larger than 10,000 base pairs. Lanes 1 to 4 were mixtures set up with the template, the synthetic primer, and anti-BSM-A1. All of them show smearing bands throughout the lane, indicating both the RCA and the nicking are successful. Also, the gradual increase in the band intensity suggests that the system efficiency increases with the template quantity. Finally, we coupled the CUHK generated from RCA to catalyze the oxidation of TMB. The mixture containing successful RCA products all showed color change with higher CUHK concentration, yielding stronger color change. As displayed here, the sample that was not cut by the anti-BSM-A1 shows slightly weaker color change compared to the cut samples. Over the summer, we primarily focused on optimizing the coupling of the RCA and G quadruplex DNA-zyme reactions. In our first iteration, we faced challenges with the construction of the RCA template, resulting in inaccurate outcomes. Additionally, this first iteration suffered from the weak signal expression. This first iteration was designed so that the G quadruplex structures could be cleaved by a DNA-cleaving DNA-zyme. However, we found out that the 13PD1 DNA-cleaving DNA-zymes did not perform well in our experiments. These results prompted us to move into our second engineering cycle to develop a more robust template. In this cycle, we designed a template that codes for the 2 repeats of the G quadruplex DNA-zyme with anti-BSM-A1 nicking sites. However, the second iteration also encountered issues during the construction, which ultimately led to unsuccessful RCA. Through this multiple design, build, test, and learn cycles, we came to our third iteration of RCA template, which is now constructed through exonuclease 1 and 3 treatment after its circularization. This version of the template shares similar features with the second iteration, except that it corresponds to only 1 repeat of the G quadruplex DNA-zyme. Overall, this version of the template provides us with greater satisfaction in terms of its performance, as shown earlier in this video. In this proof of concept stage, we mostly performed our experiments in the suggested optimum temperatures of the enzymes, which are 37 degrees Celsius or lower. In the future, however, we will focus on characterizing the third generation template, as well as verifying the coupling of the whole mechanism in room temperatures.
While Michelle is busy going in circles in the wet lab, let's do a deeper dive into the molecular dynamics of G quadruplex and heme. Since G quadruplex only works if a heme is bounded via N-stacking, it's of our particular interest to optimize the buffer in order to favor such binding. Since heme is more soluble in DMSO than in water, and G quadruplex is stabilized by DMSO, we hypothesize that the free energy of binding decreases as DMSO concentration increases. To investigate this, we have set up molecular dynamic simulations in GROMACS to obtain the free energy of binding of heme at different DMSO concentrations. We set up 2 systems, one with G quadruplex bound with heme and another with heme only. Then we gradually decouple the Coulombic interactions first, then the Lennard-Jones interactions. After that, by the thermodynamic cycle, we can calculate the free energy of binding of heme at different DMSO concentrations. As we can observe from the graph, despite the extreme data points, possibly due to changes in molecular topology during the simulation, the free energy of binding of heme increases as DMSO concentration increases. Therefore, we kept our DMSO concentration in the buffer as low as possible. While it might seem redundant to perform such simulations to arrive at this conclusion, especially with free energies that are not accounted for in our exploratory simulations, we believe that our modeling approach demonstrated how dry and wet labs could work together in order to provide valuable biophysical insights for our systems of interest. While the simulations are running, let's see how our project can impact the environmental landscape.
Moving beyond the lab, we are deeply committed to addressing the critical need in our community. To secure our project success, we conduct extensive human practice interview to understand the intricate details of antibiotic resistance gene and points of concern of implementing our kits in practice. We are aware that antibiotic resistance is not just a complex issue, but also a present one. Our human practice research provides invaluable insights and directions on probe selection, quick design, target market identifications, and outreaching educations. Some of the highlights with the doctors in Hong Kong. From esteemed doctor come executive counselor, Dr. Per Chen, to antibiotic resistance expert, Dr. Emma Ma in Center for Health Protection. These healthcare professionals emphasize the urgent need for a quicker and more affordable method for sensing antibiotic resistance. They also underscore the empirical prevalence of strong antibiotic prescription in current clinical procedures, while drawing some attention in the growing concern of AMR, such as antibiotic abuse, insufficient public awareness, in particular in agricultural sectors. We utilize this insightful discussion and consultation to shape our project in the hope of better aligning our work with the community. For instance, we follow microbiology expert, science educator Dr. Helen Ma's advice when devising strategy for educational workshop, as well as iterating and improving our wet lab mechanism after every expert interview. By investing time and effort into these conversations, the knowledge, experience, and broad perspective we gather enrich our project a lot, in particular in wet lab and educations.
Synthetic biology education has grown significantly over the years, and Hong Kong has been a part of the movement, with the emphasis of STEM education. Here at ResiSense, instead of offering generic workshops, we collaborated with community to identify the gaps in knowledge and custom-make programs for them that are specific for their needs. This can ensure that with the same amount of resources, we are able to create more impact and values. Over the summer, we created different workshops for university students, high school students, younger children, and local iGEM communities. For university students, we participated in art fair addressing the misconception around antibiotic resistance. Based on the discussion we have with Dr. Helen Ma and Dr. Factor Lau, we use interactive games and hands-on experience to not only introduce synthetic biology, but also encourage students to be an advocate for this urgent matter. We also collaborated with Hong Kong iGEM Biotech Experience, an organization that brings biotech to local students and schools. After speaking to the staff, we saw a gap in foundational workshops, specifically those for younger children. This leads us into creating a community-centered workshop, where we introduce the basic of DNA and synthetic biology through playful experiments and crafts. We believe that this approach can spark curiosity and excitement associated with the field, laying the groundwork for future learning. For local high school students, we organized a gel electrophoresis troubleshooting workshop which aligned with the HKDSC biology curriculum. By providing hands-on training, students are able to learn about the lab techniques and the critical thinking skills needed to troubleshoot experiments. In addition to troubleshooting skills and technical skills, we also partner with CityU and HKUST iGEM to share about our personal journey in synthetic biology, providing insight on the possible academic and career plan related to biotech. Last but not least, we initiated a regional symposium with other teams for the local iGEM communities. With over 100 participants, the event features presentation, poster section for networking, and expert talks on science communication. Traditional symposium often fell short due to the team's diverse speciality, whereas our event involves teaching teams how to push their project forward beyond the lab, creating an opportunity for teams to re-evaluate the social impact of their projects.
In this year, we have done a lot to give an alternative solution to AMR screening while educating the public about this issue. We hope that our project can inspire and help alleviate this problem. Massive thanks to everyone that played a part in this project, and thank you for helping fight AMR by watching this video.